Okay, so mitosis, we have our 4 stages here. Okay, up here, the way I remember it is PMAT. Okay, we have our prophase, metaphase, anaphase, and telophase. Okay, you need to know those names, you need to remember the order. Okay, the way I remember it is I just think of PMAT. I just have that phrase off in my head, so I know it. Other ways people remember it is using different analogies. So I've heard things like party Monday and Tuesday, passed my algebra test is another one. You know, you can make up your own analogy. It doesn't matter as long as you remember it. It can be as silly or as off the walls as you want. As long as we remember it, no one's going to know. Now, first stage here is prophase. Okay, so all of these diagrams, we have 1, 2, 3, 4. There are 4 stages. First stage here is prophase, metaphase, anaphase, and telophase is last here. Okay, so P is first, my prophase. And I think of that as, okay, this is preparing. I'm P for prophase and I'm P for preparing the cell. Okay, black line here, that's the whole cell. Now inside the cell we have a nucleus. So the blue dotted line here, that's my nucleus. And inside there I have my pairs of chromosomes. Now you'll also see in your notes we have a little glossary of the different terms that are coming up. So go through that glossary so that you know the terms that I'm talking about. It makes it easier then when you're describing it, okay? So here we have our chromatid pairs. Those chromatids are exact copies of a chromosome. So if you have one chromosome, towards the end of interphase, they're going to make an exact copy of itself. So we have 2 chromatids here. Okay, identical copies of each other. Inside the middle, or connecting them together, is your centromere. So this little dot here, that's called a centromere, and it's just connecting your 2 chromatids so that they stay together like that. What's actually happening then inside this this stage here, where we're preparing the cell for mitosis, the nucleus is going to start to break down. Okay, I want to make 2 nuclei, but to do that I need to break down this first one. So the nuclear membrane here, or otherwise called the nuclear envelope, that's going to start breaking down. The other thing then that exists inside the cell, so this is my cell, the cytoplasm out here, are these things called centrioles. These red X's here, just another organelle, you call centrioles. And all you need to know that they do is they're going to produce spindle fibers, okay? Spindle fibers, imagine just like a little web, okay? Like a little casting a web from these centrioles here. That's all that's happening in prophase. You've got 2 major things going on here. The nuclear membrane is breaking down and the centrioles are producing those spindle fibers. There are 2 main events. Now you need to be able to draw that diagram, so practice drawing this. With these ones, okay, it's not a case of identifying them, you need to be able to draw them. Second then, we're looking at our metaphase. So we've gone through prophase and now we're going to our metaphase M, PMAT. During metaphase here, what's going to happen is the spindle fibers that were being produced here, those spindle fibers are going to attach to the centromeres, okay, the centromeres of your chromatid pairs. Once they do that, so once they attach and line up there, they're going to line up those chromatids down the middle of the cell, okay, also known as the equator of the cell. Okay, so another way to remember that is M is for middle, M is for metaphase, okay, these are lining down the middle here. Okay, this stage starts as soon as the nuclear membrane leaves or disintegrates over here. Okay, third phase then we're looking at the anaphase. Okay, stage 3 anaphase, A for anaphase and A for apart. Okay, so what's happened here is my chromatids are lined up down the middle. They're attached at the centromere to these spindle fibers, and now the spindle fibers are going to contract, or they're pulling back towards the centriole here. And when they do that, they're going to break up these pairs and pull one of each chromatid to either side. Okay, so now we're going to have identical chromosomes at either side of the cell. Okay, it's a really short phase. They're just pulling them apart so that one of each chromosome goes to either side. Okay, our last stage then is called telophase. T is for telophase. Now, what happens here is we have our set of chromosomes, our identical sets of chromosomes on either side there. The nuclear membrane is going to reform, so we're forming a nuclear membrane around here again, and then our spindle fibres are going to break down. Now the centrioles would still be there somewhere, but they just don't have any spindle fibres coming out of them. Okay, so what you end up with here is your two identical nuclei. Okay, and once more, going back to this first diagram here, we have our nucleus dividing into two. Okay, so that's the basis here of mitosis. We're just going from one cell into one cell with two nuclei. And that's our 4 stages of mitosis. Okay, of course we haven't come to the end here yet. So we've got, we have our cell, we've got 2 nuclei in it, but now we want to divide those further into 2 cells, 2 separate cells, okay? And the way that's going to happen as well, we've got 2 different forms of cell division, depending on whether it's an animal or a plant cell. So if it's an animal cell, it's going to form something called a cleavage furrow. And what happens there is down the middle of the cell, there's just going to be a little indent is going to start forming. Okay, so this is going into my cell division here. Okay, a little indent is going to start forming on either side. That indentation, that's called the cleavage furrow, that is going to deepen and deepen and deepen until eventually the 2 cells split. Okay, and you're left with 2 separate cells. Okay, so for an animal cell, again, that's your cleavage furrow. It comes up in a question to differentiate or how do they differ. For an animal cell, it's cleavage furrow. For your second type of cell, then the plant cell, well, because there's a cell wall around it, so if I just go back here, we'll draw this back in. Because we have a cell wall around a plant cell, this indentation, that cleavage furrow can't form, okay? It's not strong enough. The cell wall is too strong to allow that to happen. So what happens instead is these little vesicles from within the cells start to build up along the middle of the plate here, okay? Middle of the cell here, and we call that the cell plate. So those vesicles are going to build up, build up, build up. They're eventually going to join with the membrane, and that's going to eventually break down the 2 cells there, and our cell wall is going to form at either side. In between that then, we have what's called the middle lamella, okay? But the main point we need to get across here is for your animal cell, we had our cleavage furrow, and for our plant cell, we're looking at the cell plate, okay? So cell plate is just those little vesicles forming in the middle here of my cell, eventually joining up with the cell membrane and dividing the cell into 2 there, okay? So there are 2 ways of just dividing the cell then, okay? And that's the whole process of mitosis and cell division, okay? That word for cell division, where we're physically just dividing up that cell into 2, that's called cytokinesis, okay? So that word there at the top of the page in brackets is cytokinesis. That can come up, or they can ask that, they might use that wording in a question instead of saying cell division. So do be familiar and just be able to recognise that term. Okay, at the bottom of the page then, of page 13, we have our checklist. Okay, so when you're going back and revising this, always refer to this checklist. Okay, these checklists are really handy. So do you know the mitosis process and cell division process inside out? As I've said before, you need to know the order. Okay, so it's going prophase, metaphase, anaphase, telophase, P, mat. Okay, do you know what's happening in each stage? So can I describe something, maybe just 2-3 events that are happening in each one of these phases here? The other thing is you need to be able to draw those diagrams, okay? So star beside those diagrams at the top of page 12, you need to be able to draw those diagrams. You need to be able to label those diagrams. So can you label the spindle fibers? Can you label the centrioles? Can you label the chromosomes, okay? Do you know what all of them are doing? What are the centrioles doing? Well, they're producing the spindle fibers. What are the spindle fibers doing? They're attaching to the centromere here in your chromatid pair there. Okay? Now, the last thing we do then in this is second last thing that we're looking at is meiosis. So we've gone through mitosis in great detail. Now, you might be panicking and thinking we should do the same with meiosis, but we don't, okay? All we need to know for meiosis is what it is, okay? And remember, we said before that meiosis is going to have 4 daughter cells. We're going to have half the number of chromosomes in each of those daughter cells, okay? The other thing we need to know is its role, so what's the role of meiosis, and then just being able to differentiate between meiosis and mitosis. So again, we have our function then is so that if we have 2 haploid cells, then we can allow for 2 haploid cells to fuse together and form another diploid cell. So that's important in reproduction. As well as that, it's going to allow for variation. So if we have the gamete of one individual and the gamete of another individual, they're 2 different people joining together, it's creating this new individual different from either both parents, okay? So slight variation of parents, and that keeps going. So that's why we're all so different. We've got variations from different parents. Now, in that table then on page 14, you just have some brief points on your difference between mitosis and meiosis. In general, you'd usually be asked for about 2 differences between mitosis and meiosis, and that could be something like a 6-mark question. Nice, simple 6-mark question in one of your short questions. So you could just say, right, mitosis has 2 daughter cells, meiosis has 4 daughter cells. Mitosis, remember, we have the same number of chromosomes in the daughter cells, and then meiosis, there's just half the number of chromosomes, okay? Last topic then that we're going to discuss today is cancer, okay? So, what is cancer? It happens when it's a group of disorders where the rate of mitosis is lost, okay? So normally in our body cells and all of our cells, the rate of mitosis is clearly and carefully monitored. So the cells are only dividing when they need to, okay? Cells are only dividing when they need to grow and to repair some certain cells. What can happen, and we're not really sure why, but what can happen is the rate of mitosis just loses, it's lost control. And what's happening there is you've just got lots and lots of these cells being produced over and over again, so you end up with this mass of cells, okay, resulting from this uncontrolled mitosis, and we call that a tumour. Now, not all tumours are bad. I know we live in a world where cancer is really scary and really petrifying to a lot of us, but not all of those tumours are bad, okay? With some of them, it's just there's going to be lots of mitosis, it's dividing over and over again, but it's not invading any tissues, it's just kind of chilling there, and it's not causing any harm to any other part of the body, okay? That would be called a benign tumour, okay? The word benign meaning kind or friendly, so they're just sitting there in our body, not making any difference or causing any harm to our bodies. The troublesome cancers then are your malignant tumours. So what happens there is those cells, they divide by mitosis over and over again, forming that big mass of tumours, and instead of just sitting there and not bothering any other cells, it's going to start invading other tissue in the body. Okay, that can then move around the body, infecting other areas of the body, and that's where the trouble starts, really, essentially. So from that, you just need to know your definition of cancer. So we have our definition there. It's a group of disorders where the rate of mitosis is lost, okay? So uncontrolled rate of mitosis. Causes of that then, so we could have causes such as carcinogens and genetic mutations. We go further into that then when we do our genetics again, okay? So just going back again over what we've covered in this chapter here, this is one of the major chapters that comes up in your short questions. So we've covered our functions of cell division, and remember, you need to know the difference whether it's a multicellular organism or a single-celled organism. Our chromosomes again, so that's talking about our DNA and our protein, just what are they made up of? Cell cycle then, so that's cell cycle, it's the lifespan of the cell. Remember, all it is, it's either just going to be dividing or it's not dividing. And mitosis and meiosis, one of the major parts coming up in short questions. The only years it hasn't come up is 2012 and 2010. Okay, that's since 2002, so just goes, or 2004, sorry. That just goes to show how vital and how this is one of the main sections in the Leaving Cert here. Can be anywhere usually between 5 and 6% of your Leaving Cert. Okay, so again, just making sure you know this inside out. Keep going back over, going through that checklist. Your last thing then is cancer, so knowing your definition there of cancer, and that can come up as part of your short questions, maybe in terms of mitosis, or if you're looking at interphase or meiosis. Okay, so at the end of these notes, we also have some of the exam questions. I've included all of the exam questions that have come up to do with mitosis. So I just want you to look at page 15, and this was the most recent exam question that came up in mitosis, and it's looking at the different stages of mitosis, but it threw some people that year, because what we're looking at here is a diagram of the cells under a microscope, as opposed to an animation like this, which most students would be looking at, most students would learn. Okay, so what you have here, the questions that are asked, is which type of microscope is used in the school lab to observe cells like this? Now that's going back to our cell structure question, and remember the one that we use is our compound microscope. Okay, the first cell is in prophase, so in that diagram there, so just make sure you're following along while I'm going through this. We have, first cell is in prophase. Give 2 observable events that happen during prophase. So remember prophase, think, okay, we're preparing the cell. Okay, we're preparing for this split to happen. So what was happening there is we had 2 things to prepare. Our nuclear membrane is going to break down, and then secondly, our spindle fibers are going to be produced. Okay, you could also say about the chromatids here that the chromosomes duplicate or replicate, forming your chromatids there. Now on the photograph, use the letter M to label a cell that is undergoing metaphase. So remember our metaphase, think M for metaphase, M for middle. So what you're searching for is a diagram, okay, some diagram here where it looks like the chromosomes are lined down the middle. If you look at that diagram, there's only one there, so it's the third diagram, third cell from the right, and that's where all the chromosomes are lined down the middle, or down the equator of the cell. Okay, then we're asked to label A, which is going to be anaphase. Remember, A for anaphase and A for part here. Okay, so you're looking for a stage where the chromosomes are being pulled apart. Okay, and that looks like it's going to be the second diagram from the left there. It's going to be our A. So after mitosis, the cell divides in 2. Again, our term for that is cytokinesis. This happens differently in animal cells and plant cells, and then describe what happens in animal cells and plant cells during this stage. Okay, that year the marks were given for animal cell, you just needed to write cleavage furrow, and then for plant cell, you just needed to write cell plate. Okay, you got your 3 marks and your 3 marks there. All of these questions, okay, so it's going all the way back to 2002, mostly short questions, but you do have some long questions as well. The marking schemes for all of those are in the back of the book as well, and what you'll find is that with most questions throughout these Leaving Certs, and it's good to give a look at these marking schemes, you find that most questions are worth about 3 marks each, okay? So every percent, or every 2 questions that you do, that's working up to be 1.5% of your exam, okay? And that's a good way to look at it when you're going through these exam questions. Start off simple, going through what you know, and then working your way up, okay? So just to summarize then what we've gone through the cell structure, we've gone through cell division, okay? Throughout the year, what I would be doing is focusing a lot. For 5th year, I focus on a lot of Unit 2. So that's things like photosynthesis, respiration, genetics, and cell division. Those are really confounding kind of topics in the Leaving Cert. They're coming up time and time again, and they can be helpful then to you in 6th year when we go on to do your Unit 3. Okay, Unit 3 is your different systems, so we got our different human systems such as, you know, breathing, digestion, excretion and all of those. We also look at plants in great detail, and as well as that, your microorganisms, okay? So just, I didn't mention it before, but we will go back to Unit 3 there, and we'll just look at how all of these topics here. So we've got our bacteria, fungi, protista, viruses, then we have the plant, we've got the human side of it as well, okay? We've got all of these chapters. It does seem quite daunting, but the way I would look at it is I break it up. So up here, my first four topics here, they're going to be my microorganisms. Once I get those locked in, okay, they're fine and they're okay. Again, you need your knowledge of cell, enzymes and cell division osmosis here. It's really going to help you when we go to do this Unit 3. The second thing that we're looking at is the plant, okay? And again, your plant, we're talking about structure, transport, responses, reproduction. Lastly then is the human anatomy, and that's going through all of these systems here, okay? Unfortunately, it can be a lot of detail here for quite few marks, okay? Or sometimes unpredictable topics can come up. For that reason, I think it's really important that we focus on Unit 2 and Unit 1 for 5th year, and then once you get to 6th year, you know, these kind of all slide into place and they'll become more more relatable and more easy to understand, okay? Okay, so that's the bones of the course there. You know, we're going through our exam, and again, we're focusing here at the Academy, focusing on the exam, trying to get you into that mindset of answering those questions the way the examiner wants the questions answered, so that you get the most out of your marks. Throughout the year then, I'd be hoping to cover between 60 and 70% of the course. You get notes on all chapters. We go through most of them in class. There'll be some that you'll just be given, you know, the shorter kind of more autonomous chapters that you can go through yourself. We're here every Tuesday evening throughout the year, so it's here in Stillorgan in the Dublin Academy of Education in Stillorgan. For more information, you can just contact the school about those. So I look forward to seeing some of you here in September. Thanks.